Nirenberg and Mattaei, the poly-U experiment and their contributions to genetics. Nirenberg and Mattaei were the first to recognize that specific RNA sequences coded for a specific amino acid. They determined this groundbreaking discovery using an experiment known as the poly-U tube experiment. In order to emulate all components of a cell without the confinement, a cell-free extract was used, meaning everything was taken out of the cells and placed in a tube. The cell-free extract was combined with a string of U's called poly-U mRNA and 20 amino acids. One of the 20 amino acids was radioactively labeled. This procedure was repeated 20 times, each time tagging a different amino acid. In performing this experiment, the scientists aimed to discover if the radioactive amino acid would be incorporated into a protein. Nirenberg and Mattaei noted unusual activity in one of the test tubes. A chain of uracil bases produced a protein chain made up of a singular repeating amino acid. Thus, they concluded that the poly-U directed protein synthesis, or in other words, the poly-U coded for phenylalanine. They repeated this experiment with a poly-C, a poly-A, and a poly-G. The poly-C coded for proline, the poly-A coded for lysine, and the poly-G did not work. Nirenberg and Mattaei made significant strides in cracking the genetic code. Nirenberg, on the left, continued on to work with Lieder in hopes of discovering more about the genetic code. Using a combination of short mRNAs with different codon combinations, tRNAs, and ribosomes attached to amino acids, the scientists hoped to answer the question, will the radioactive amino acid bind to the ribosome? The experiment was repeated multiple times, tagging different radioactive amino acids. If binding occurred between the tRNA, the ribosome, and the codon, then it would get caught in the filter paper. If the tRNA with the radioactive amino acid did not bind, then it would pass through the paper. Nirenberg and Lieder were able to conclude the specific nucleotide codon sequence of the amino acid caught in the filter paper. By 1966, Nirenberg and the other scientists had translated the 64 RNA three-letter codons for all 20 amino acids.